right, I think we're just about ready here. Someone can confirm for me.
Yeah, we're
Okay,
good.
Wonderful, wonderful. Okay. Okay, good afternoon, everybody. And I want to take this opportunity to say welcome to what I believe to be our 11th chat with the frat, sponsored by the Brothers of Alpha Phi Alpha Fraternity Incorporated, Omicron Phi Lambda chapter here in South Fulton, Atlanta, Georgia. My name is Dr. Gary White. I'm assistant professor of social work, practitioner and scholar and community activist with Clark Atlanta University. And I'm excited for what we are going to unpack and share with you all on today. As many as you know, on last week, this represents really part 2 of our discussion around vaccinations. Last week, we began by putting it in a historical context in which we recognized and identified some of the historical concerns that many people of African descent had with regard to participating in clinical trials, as well as with regard to participating in vaccinations. And so this is particularly exciting on today. And what makes it exciting is because we had to address that historical concern around that clinical trials and vaccinations. And the brothers did an outstanding job in engaging in that communication. We also recognize that that was important because it set the framework for the discussion on today with regard to actual vaccinations. And so I'm going to invite everyone who's watching this to get someone online. Any questions or concerns you may have about vaccinations or clinical trials, now is the opportunity when you see who we have organized to address this issue from the perspective of African American men. And so, and all of these medical doctors that you'll see and researchers that you're about to be exposed to, they recognize that we are in an unprecedented time, a time in which clinical trials are happening at a pace that has not been done before. Something as some medical doctors are going to share with you today, it is not within the context of the typical time frame in which a vaccination is brought forth to the community. And so we're going to have an outstanding time on today. And so without further ado, allow me to take this time to first introduce to you all the members of the interdisciplinary team, followed by our guest speakers on today. The members of our interdisciplinary team are Brother Marcus Dumas, public health expert, Dr. Christopher Bass, a clinical psychologist, Dr. Leroy Graham, medical doctor, Brother Kyle Bryant, a doctoral candidate in risk communications, Dr. Darren Wright, associate professor of social work, scholar, and community activist, and also Brother Henderson Johnson, also a doctoral candidate at Georgia Tech University. That's our rocket science man. And so allow me now to take this opportunity to bring forth to you all our guest speakers on today. Some of the, as well as as well as Dr. Terry Dickerson. So allow me to introduce our guest speakers today. And I'm going to ask our guest speakers to give us your name, and as well as very briefly your medical background so that our viewers may put in context and be able to more appropriately direct their questions. So I'll begin with you, Dr. Dickerson.
Yes, I'm Dr. Dickerson. I have a background in doctorate in podiatric medicine, board certified foot, ankle, and foot and ankle surgeons. Also have a board certified in laboratory medicine with American College of Clinical Pathologists, and I also have a Juris Doctorate degree as well.
Thank you very much. And we also have Dr. Oliver Richmond.
Good evening, brothers. Good evening, guests. So my name is Dr. Oliver H. Richmond III with the Eta Lambda chapter of Alpha Phi Alpha Fraternity Incorporated. I am currently a research scientist for cancer biology at the National Institutes of Health and the Heart, Lung, and Blood Institute in Bethesda, Maryland.
Beautiful. And we also have interdisciplinary team member, Dr. Leroy Graham.
I don't even play the bass. I'm Dr. Leroy Graham. I am a pediatric pulmonologist and did pediatric pulmonary and critical care, also an Army veteran, former brigade surgeon in Operation Desert Storm. I am passionate about the disparities that we have, and I'll just start right there with, as of 6 p.m. Eastern Time, 6,967,910 confirmed cases of coronavirus. In the United States, we are number one in the country with 1,931,325 cases. The nearest country to us is now Brazil at over 672,000. In terms of deaths, there are 401,368 deaths worldwide. The US is also number one in that category with 110,110, followed by the United Kingdom at 40,625. COVID is an insidious pathogen, as many of the people will discuss. One of the things that we will hear a lot about it is an RNA virus, which is a particularly vile infectious agent and invades our own cells, where it takes over manufacturing of proteins and thereby signals messages to our own cells to reproduce itself. Literally, the New York Times says COVID hijacks our cellular process.
Stop right there. Hold it right there. Keep it going. I can't wait to get to you, Dr. Graham, so that you may finish that. We don't say 5 minutes of funk, but we say 5 minutes of facts. So I'll get with you in 1 second. Allow me to finish introducing the other guest speakers, and then we're going to move into it. But please keep those facts, Dr. Graham, because I'm sure our viewers need to truly understand it, to put this pandemic in perspective. But very quickly, I want to introduce Brother Dr. William Bennett.
Good evening. I'm William Bennett. My background is in surgical and anatomical pathology. I spent probably the last 20 years working in the pharmaceutical/medical device industry. My primary function at this stage in the game is to try to develop the next best therapy for patients who are suffering from all forms of cancer. So I'm happy to add my perspective to this discussion tonight and help to lead some of these discussions as well.
Thank you very much. And finally, our last guest speaker, all the way from the West Coast, who will also be adding color commentary to this dialogue, Dr. Bouchard.
Hey, good evening, everyone. My name is Dr. Esteban Bouchard. I'm a professor at the University of California, San Francisco. My expertise is pulmonary and critical care medicine. My scientific expertise is studying racial differences in risk of asthma in minority children and lung disease and drug response. Thank you for inviting me.
Thank you very much. And so certainly, as you all see, we are very excited and delighted to provide with you some of the top brains to address this issue. We call it a shot in the dark in the race for the cure around vaccinations. And so to do so, hold it back up again. I missed that. What was that?
White coats for Black lives.
White coats for Black lives. We're going to make sure, look at that. We're going to make sure we double back on that, because that in of itself is a movement. So keep that in mind. I love the way he threw that in there. And I'm going to make you pull it back up again later on. So let us go right into the discussion and dialogue without further hesitation. Once again, everyone who is viewing this, if you have any questions or concerns, please on Facebook, hit us up. We'll bring those questions into the room. For those of you who are present on here, again, any questions or concerns, throw them in on the message group, and we'll make sure that the team brings those questions into the dialogue room. All right, so let us begin by a quick recap. Like I said last week, we began with addressing the issue of the concern and the anxieties that many people have around participating in the clinical trials, as well as around participating in these vaccinations that may come forward at unprecedented levels. So we're going to start this conversation with Dr. Christopher Bass, clinical psychologist in the Department of Social Psychology at Clark Atlanta University. Brother Bass, if you will, please give us a recap on those issues and concerns, which again laid the foundation for this particular discussion.
Thank you, brother Dr. White. And you did an excellent job already discussing what the brothers talked about last week. But let me say where we finished. Where we finished, we really began to examine how are these anxieties learned? Why do we come up with these fears? Dr. Wright did a wonderful job last week, really breaking down the historical context of what happened previously as it pertained to clinical trials and experiments and so forth. So I wanted to start today before we really get in going and come back and do a full circle as it pertains to anxiety and fear. And I'll say something here that most people understand, but we really don't talk about a whole lot. Anxiety is really based on the unknown. Fear is based on what's known. So when we talk about anxiety, it's really hard to have stable footing when we continue to get information from all different sources and then find or come to learn that many of the pieces of information that we get is wrong or, scaled a different kind of way. We had a discussion a week ago or 2 weeks ago looking at the numbers and how the numbers sometimes present a different type of story. So going back 3 weeks ago, I need to sort of bring on Dr. Wright, or really talk about what Dr. Wright mentioned when he said the Sankofa moment. And Sankofa, as he described it and educated us all, meant to go back and get it. Today, we have an understanding or opportunity to go back and get it from those people who actually know this science. We're going to learn factual information today, and hopefully with full information, we won't have the same level of anxiety because we'll have something to stand on. So again, anxiety is based on the unknown, but hopefully at the end of the discussion today, we'll have some things that are known. That's it.
Thank you very much, Dr. Bass, for putting that in context. And again, hopefully the original discussion that we had lowered that level of anxiety and reduced the level of fear concerning that which we do know in terms of its impact. But today, hopefully we're going to impact and reduce that fear even more with the facts and the figures and the information that the doctors are going to bring forward. So we're going to go right into this discussion right off the bat with regard to really just trying to make a fundamental distinction between vaccine versus treatment. Putting a context in terms of how these two concepts differ and exactly what it is that we really need to know with regard to how these two differ. So I'm going to start with you, Dr. Graham, and as I said with the other medical doctors, please feel free to add additional commentary to each of the points. Dr. Graham, you're on mute.
That is an incredibly important distinction. When we speak of therapy, when we speak of the number of therapies that have been advanced for COVID as treating a disease, much like you would treat an infection or another condition. We talk about vaccines, which may be the ultimate hope in this disease, as has been with other coronaviruses like influenza. We talk about giving purified extracts or proteins, training our immune system to recognize critical parts of the virus going forward. Armed with that, if we are successful, we can contain this disease. So that is an important distinction. And before I go on to the other speakers, I would just like to say that it is so important that we get minorities involved in these studies, because the issues of pharmacogenetics and pharmacogenomics, which I think will be dealt with much better by the following speakers, are so very critical. We must be included in these studies for these possible vaccines and these possible effective treatments to be of greatest benefit to people who look like us.
Powerful. So very quickly, so what happens? And I know I heard Dr. Bennett mention it last week, so I know he's going to double back on that again. But for you, Dr. Graham, fundamentally, what happens if we are not represented well in the clinical trials? What does that do with regard to the benefit of any vaccines with low involvement from our population?
Well, both in terms of treatments, actual treatments, and the vaccine. Examples, as one of our other speakers that I've done some work with can elucidate. You take a disease like asthma, okay? The treatment for asthma, as Dr. Burchard has shown, is basically and fundamentally different for African Americans. And so African Americans are treated with a therapy that has not been studied in them. It's not as effective. There are some nuances and so forth. The same thing will apply to this virus and to the vaccines. If we are not adequately represented in the studies that prove the efficacy of the vaccine, or more importantly, even the therapies for people that become seriously sick with this, then these therapies will not be applicable and will not thereby be of benefit to us. So it is very critical that we be represented. And I think, as other speakers will say, there is a sad history, as we discussed last week, of we as a people being misused in this context. And we have to get beyond the fear of that, learn what these studies are, learn the values, and in an informed consent, which is what ethically and legally should be done, involve ourselves so that the results of these studies are reflective of our uniqueness.
Beautiful. So, Brother Bouchard, Dr. Bouchard, he mentioned you. So I'm going to just raise a question. What are we fundamentally getting wrong with regard to these distinctions about a vaccine versus the actual treatment or the cure?
Sure. Before I start, I noticed that Dr. Erica Ferran is on. She's a young African-American female lung specialist at UCSF, and welcome, Erica. What you talked about fear versus anxiety, and there's real fear because of Tuskegee, which ended, with 1972 as a federally funded research study to look at the natural course of syphilis, where they gave and injected African Americans with syphilis, and then just wanted to see the natural course and didn't treat them. When they gave it to white folks, they treated them. So that is a fear, not an anxiety. It's a true federally funded observational study that ended within my lifetime. With respect to treatments, 1993 is a pivotal point because Bill Clinton put into law that it was required that all federally funded research include women and minorities. And that law was implemented in 1994. And we're coming up way past the 20-year anniversary. And when we look at other studies like cancer research, less than 5% of all clinical trials included minorities. So that means that if you're not participating in the clinical trials, you're not understanding what are the different risk factors for cancer pertain to your population. And we see that triple negative breast cancer is the most common form of African Americans, and yet we don't have any therapy for it. Part of that is because African American women either chose not to participate or were not included in the clinical trials. Now, it's been over 50 years since we've had HIV and we don't have a vaccine yet. I'm not too optimistic that there's going to be a vaccine for SARS-CoV-2, but we have to try. And we have to make sure that we have minority representation in the clinical trials. And we have to have minority physicians and scientists at the table designing those clinical trials and making sure that minority folks feel welcome and are included, and their welfare is taken care of and monitored while they're participating in the clinical trials.
And,
Wow,
and Dr. Watt, if I just may add.
It takes us, Dr. Bennett, it takes us right to you. This is like an alley-oop, Dr. Butcher. I'm moving that right on to Brother Bennett. Dr. Bennett, I want you to add to that. And then if you will, as I listen to them, Dr. Bennett, I want you to also add to that and then help us understand a little bit about the clinical trial development process.
Sure. So Dr. Bouchard brings up an important point. There has been a fundamental shift from the early 90s to where we are today. Prior to that, the presence of African Americans being as a part of a trial design has always been somewhat absent. And a lot of that went back to, again, the Tuskegee experiment, but you can go further back and see where there has been a number of different atrocities involving people of color involved in trials where they've been mistreated in one shape or another. And so there is a fundamental fear, it's a rolling fear that happens from year to year. And I do agree that, and Dr. Bouchard, hopefully this is not a surprise to you, but I do agree that from my vantage point, there may be some challenges in trying to develop a vaccine for COVID-19. I am not as optimistic as many people are in a situation like this, because what has been a scientific process prior to COVID have now become a political process. And so the trial designs now have essentially been thrown out of the window. And so you have politicians that are speaking in terms of what I would consider junk science, to be honest with you. I believe the mumps was the last vaccine that we developed that took roughly 4 and 5 years. And here we are with COVID going to maybe 7 months. And here we are talking about developing a vaccine by year end. That is virtually impossible.
So, Dr. Bennett, you mean to tell me when they got on and said that we were going to have a vaccination within a year, so are you telling me that you weren't the only medical doctor whose eyebrows went up?
I think the whole medical and scientific community was probably in a farce at that point, because we understand that there is a process that must be followed, because you have to ensure that 2 things are met. Patients need to be safe in those trials, and the therapy that you're developing must be safe, and it also must be effective. So let's just dive into sort of the different processes, if you indulge me, as far as how a therapy essentially goes from the benchtop to bedside. And in medicine, that's the pendulum of the drug development process. So you have the discovery and the development phase. The discovery and the development stage is something that happens in the lab. These are things that generally do not involve humans. They may involve human-like cells or cells derived from human, but they're not involving, say, for example, a person being involved in a particular trial. Those are the basic stuff. These are the things that takes years. These are the things that may take anywhere from 10 to 20 years to develop what we call a good compound, so to speak. And I'm just using that word quite loosely here. So that is that particular process. That's the initial phase of trying to get that part of it to what we call solid science, so you can get into a much more robust discussion on development in what we consider the preclinical research. Now, the preclinical research is, and again, if you're an animal rights activist, it may be somewhat controversial, because the preclinical component does involve animals. This is where you have to develop a pretty good animal model where you're testing that particular compound that you have derived from sort of the discovery phase to ensure that this particular compound actually works. So before you even get to the stage where you're designing a trial, and I'm sure many of you have been hearing on TV lately about, randomized trials and open label trials and those kinds of things. Before you even get to that stage, you really need to make sure that you have crossed all your T's and dot all your I's, that particular compound is effective and safe in your animal models. And so once you've essentially exhausted that process, and let's be clear, the preclinical studies takes years. This is not something that happens over months. It takes years and a lot of investments to be able to push that particular compound to a stage where you can now write a protocol, submit that protocol to the Food and Drug Administration, and the Food and Drug Administration gives you the green light. So now you're able to execute what we call a phase 1 trial. So a phase 1 trial is generally a very small, controlled trial, all right? Now I want you to think about that, because what you've been hearing lately around the COVID-19 and these large trials that they're essentially conducting, those aren't phase 1 trials. Phase 1 trials are essentially very controlled. They're small, anywhere from 25 to 30 people. They're monitored very closely to ensure that that therapy that you have derived from the preclinical model is not only safe, but it's also effective. But there is a surveillance that happens over time, even in that phase of the drug development process, because I'm going to use sort of some elementary terms here. So you don't want to give a therapy, all right, and a patient essentially starts to feel good 30 days after you give the therapy. Well, what happens 9 months down the line? What happens 12 months down the line? There has to be a system built into that process where you are ensuring that that particular disease or virus patient doesn't go into relapse, right? So you have to monitor those patients for a while. And that phase of process can last anywhere from 2 to 5 years. So if you add all of that up, it is not a quick process, all right? And then once you've exhausted sort of that phase 1 process, and the phase 1 process does include somewhat of a subset of phases. You have what is called the dose expansion cohort, and you also have a dose escalation cohort. And that's where you're essentially determining, all right, I want to develop this drug for COVID-19, and I want to make sure that 5 milligram of this particular therapy works. Well, yes, so this expansion, you want to expand your dose. You want to test different dosages, 5, 10, 15, 20, whatever the case may be. So you want to find the optimal, that's what they call the optimal dosage, because you need to have a optimal dosage essentially established before you get into your later phase of your trial. And then you also want to go to what we call an expansion phase. You want to expand your pool of patients, including the therapy in that phase of drug before you get into a phase 2. Now, your phase 2 component of it is what we call a larger cohort of patients. These could be in the hundreds of patients that you're essentially putting on a particular trial. And again, we haven't even taken in consideration yet the process as far as, do you want this study to be an open label trial, or do you want it to be a double blind trial? Do you want it to be a randomized trial? And so without getting into the specifics of those different designs, they are important, because when you're developing a trial, what you need to do is to rule out biases in your trial. So oftentimes that's what you see in randomized trials and you see in open label. But at the same time, one of the things that Dr. Graham mentioned early in his discussion is the process for including African Americans in trials. So you have what is called a stratification factor. Stratification is a series of different elements that have to be built into the statistical baseline of that trial to determine a good power for your trial, all right? So let me give you a couple examples. So if you're developing a asthma therapy, and you already are aware that the vast majority of patients that are affected by just asthma in America, period, are African Americans, would it make sense that you have a large pool of African Americans in that trial? Because again, Dr. Graham mentioned this earlier, that you may put a drug on the market that excluded people that look like you and I, and the therapy is not as effective as those other folks that were in that particular therapy, in the actual initial clinical trial. So gender, race, ethnicity, regions of the world where that patient may live in, it could be performance status. There's so many different elements that you have to build in up front, and you have to make sure that when you execute the trial, you take all of those things in consideration. And that's the phase 2. Now, phase 3 is what we call the more pivotal component of your trial. If you've made it to a phase 3, that means you've gotten clearance from the FDA. Now think about this. Let's forget about the discovery phase, which is the initial phase, and start at your preclinical until you get to your phase, the initial start of your phase 3. That's anywhere from 5 to 10 years before that probably happened, with millions of dollars invested in the process like that. This is no quick and easy thing. So when you're hearing all of this, we're going to have a vaccine by December, when you just discovered a virus, which is novel. You've never heard of this virus before. It is brand new. We know very little about the etiology of this particular virus. It's been studied it's not been studied at all. And so I'm not optimistic. So, Dr. Rashad hit the nail on the head. This is where facts in science come into play. A phase 3 trial, go ahead.
No, go ahead. Please go on. Please go on.
No,
Well,
so
I wanted to add to Dr. Bennett's example about the asthma. Here's a package insert for one of the first big drugs that came out for asthma. And, every one of us gets this package insert, and we throw it away. It's the first thing we throw it away. And this is why you throw it away. But on this particular page, I have to stand up to show you guys. This is the first, see, it's labeled and has a big arrow. This is for Ceravent. But if you read the small text, which you can't read, it says if you're African American and take this medication, you have a 7-fold increased risk of dying.
Whoa, hold.
Yeah, you can't read it. Even I can't read it with the magnifying glass. But this is in the package insert. And this is to Dr. Bennett's point. If you ain't at the table, you're on the table.
yeah.
Dr. White,
Dr. Dickerson, go ahead.
Yeah, I would like to add to what my other colleagues are basically saying. The magic word is the FDA has created what you call the EAU. That's emergency authorized use. So therefore, what Dr. Bennett and Dr. Bouchard is referring to going through these trials, and also Dr. Graham, unfortunately, they have kind of eliminated those barriers. And so they have allowed the companies and the research institutions to shorten these trials to basically kind of what you would say sidestep the time frame that you normally would take 3 to 5 years in phase 3. They have allowed them to shorten it with their approval. So that's what's going to make this particular vaccine somewhat unique, because some of the barriers that they normally in the intense trials that they normally go through, those trials time frames has been shortened due to the label of the emergency authorized use for the FDA. And also, I just want to simplify things for people that's out there that's kind of not medically astute. Basically, when you're dealing with the vaccine is basically being created for someone that's you trying to keep a disease from occurring. So that's what the vaccine is basically for in layman terms, or if we haven't mentioned that. And also, when you're dealing with a treatment, the disease is already there. And so now you're trying to deal with that particular disease. Now, when you're dealing with a cure, now the disease is there. Now you're trying to eradicate that disease, period. So that's just for lay people out there didn't understand the difference between the vaccine, the treatment, and the cure. So now you have it.
Okay.
Yeah, so if I just add one thing to Dr. Dickerson, what you just mentioned around the authorization use. Now, let's be very clear. The EAU it's permissible, and again, global health authorities have to grant that access. Here in the U.S., it's the FDA. In Europe, it will be the EMEA. In China, it will be the CFDA. So those institutions would essentially grant that. But you have to recognize that there is an inherent risk when you're shortening the time span of the development process, because although as a drug developer, you want to build in as much safeguards as you possibly can into that process, the challenge with that is that you don't want to rush the process of baking a cake. You need to make sure the process essentially play itself out over time, because there are things that will, quote unquote, develop and in the process of you developing the therapy that can confound your results, and you get a lot of false positives. And that's the challenges that you run when you shorten a particular process in the drug development process. So Dr. Dickerson is right on this, on that point. It's just that as a researcher, we want to exercise as much due diligence that we possibly can up front to reduce any downstream harm that it may cause to patients and also the short-term harm that it may cause to patients. And
Can I jump in on that one?
Yes, go ahead.
You know,
Hold on. Give me
today
1
is-
second. Hold on
Sure.
1 second, Bouchard. Y'all are flowing. This is like a dream come true in terms of the flow. I don't want to disrupt the rhythm. I do want to do this very quick. I'm going to get back to you. I want to hear very quickly from Dr. Richmond and then Dr. Bennett. I want to make sure that you don't leave out that third phase that I'm sure that Dr. Dickerson and Bouchard are going to add to as well. But very quickly, Dr. Richmond, given your particular background, did you want to add to that?
Yes, I just want to reiterate a couple of things briefly before I'll touch on the rest later in my presentation. But so first is, so the timeline proposed publicly for the development of these potential treatments and cures or vaccines came from one person that happens to head the Oval Office of the United States right now. So I just want to put that out there. No self-respecting scientist, to my knowledge, has gone on to any publication or any news outlet and shared that information, because it's unfounded. It's not factually possible. And so with that, this is a great opportunity for everyone to just do your own homework and don't idly sit back and just passively absorb information that's given to you, whether it be your favorite CNN or Fox News channel. The other thing is as Dr. Bouchard mentioned earlier, if you're not on the table, then if you're not at the table, then you're on the table. But a way to get to the table in order to shape and affect the conversation is to be a part of the design and development of these clinical trials, to help with the thought process. There are questions about you and I and our sisters that we can develop or that we can pose that directly affect us that our counterparts may not even think about. They may not be inherently racist. It's just that from their understanding and from their experiences, which are different from our own, they don't think about those things that affect us. And so if we want a drug that's going to come out here and treat this prostate cancer or treat this triple negative breast cancer, which proportionally or disproportionately target us, then we have to have a seat at the table. We have to be part of that conversation.
Wow, thank you very much. So very quickly, I appreciate you. Brother Bennett, I'm gonna go to you, and then I know Dr. Bashar wanted to add to that, but very quickly, a quick recap. Phase 1, you referred to as discovery phase.
Mhm.
Phase 2, you referred to as what?
So again, the phase, you're talking about the full gamut of the process. So you have
Yeah.
your discovery and development phase. Then you get into your preclinical models. The preclinical models involve animal testing. Then
Okay.
you get into what we call the clinical trial phase. Now, the clinical trial phase includes both phase 1, phase 2, phase 3, and then you get an FDA approval. And then after that, you can go into a phase 4, which is your post-marketing study. So we've already talked about the discovery set phase, your preclinical phase, your phase 1s, your phase 2, now your phase 3. Your phase 3 is where you are looking at examining a larger pool of patients. All right. And in this phase, you can do what is called a comparative analysis as well. Does this particular drug more effective than this one, or is this one more superior than this one? So those are the kinds of things that you're challenged with in the phase 3. The great thing about a phase 3 process is that you're almost there. You can almost see the light as a developer. So if you can demonstrate your primary and secondary endpoints, which is built into your statistical consideration from the very first part of the trial, then you can essentially develop this beautiful dossier that you submit to the global health authorities and say, hey, my drug works. I would like to apply to be able to market this therapy amongst wherever. And so that's the pivotal stage. Now, this can take, again, years. A phase 3 trial can take 3 to 5 years before you can actually finish what we call your last patient, last visit. All right. That can take a long time. And then once you get that, then you go into the other phase, which is the waiting game. You submit your dossier before the FDA, and at some point you'll have to go in front of the FDA and essentially present your data. FDA gives you an approval probably a year or two after you've submitted your application. And then at that stage in the game, the trial process is not done. You have to go into what is called a post-marketing research phase. And that's your phase 4. That's once the drug is on the market, folks are starting to take drugs, people are living or dying, then of course you have to go into that stage. And that's where the process continues.
So very quickly then for you, Dr. Bennett, before I quickly go to some follow-up comments with Dr. Bashard and then receive Dr. Richmond's communication. So for people watching this, the elderly and other folks who are watching this, and so they receive this notice. So therefore, based off of what you've eloquently articulated, if they're getting invited to participate in clinical trials, based off of what you've now said, we know that they're, whatever this medication is, they're in possibly phase 2 or phase 3 in this process. Is that accurate?
Well, it depends, because you can invite them into a phase 1, 2, or 3. It all depends on what stage and whether or not they're qualified for that stage. Because remember, there are eligibility criteria that you have to meet in all of those different phases.
Okay, gotcha.
So yes.
All right. So, and that's important too, because again, within the context of the when and where I enter in this, again, I'm operating on the premise, and for all of our great doctors on here, I'm operating on the premise that you have people in the African American community see this call, see these postings and notifications for participating in clinical trials, or their doctors whom they, for all intents purposes, see as God and the final word. And so they're listening. So understanding where they fall in this or where the medication is critical. One point I am going to put on the post-it board before we get out of here is I'm going to ask your specific challenge in terms of a patient of you who trusts you 1000%. Before we end this conversation, I'm going to ask each of you medical doctors, how do you explain to your patient about the benefits of this and you don't fully know? but we'll get to that before we leave out of here for today. Dr. Bashard, I know you had something to add, and then we're going to go to Dr. Richmond.
Yeah, thank you. I just wanted to echo what Dr. Bennett had said earlier, is that the timeline is very optimistic, and it's being put out by the head of a reality TV show who happens to be head of the presidency right now. And we've had HIV since the 1960s, and we still don't have a vaccine. So the reality show CEO has kowtowed the Fauci and the head of the CDC, and he is unrealistically putting out timelines because he only has 5 years or 5 months of his presidency left. And so he's really raising the stakes on unexpected realities. And that's the reality. I'm not optimistic. We're not going to see it by 2021.
Well,
Bashard,
and
I
I'm
just
glad
find it
you said.
interesting. Dr. Bashard, I was just going to mention, I said, I mean, don't you find it interesting that when you have our experts, our infectious disease experts, outside of sort of the noise, and when you ask them, one of the things that I think they have a moral obligation to disclose to the public is walk the public through what's involved in developing a particular drug and let folks know the truth. I mean, sometimes
if they don't get fired first.
exactly, and that's what's been happening. You're right.
Well, I'm blown away by the degree to which you all are being honest, because as Eric Morgan put on the post, something Dr. Bashard that you just said, and that Fauci a few days ago said that by 2021 we should have several vaccines that have come forward. And so someone whom we trust since he hit the scene in the 80s with HIV/AIDS, he's providing that information. So that's why I'm so glad that I have you all here, because our people are hearing all different types of information. And so it's going to be critical that you all take a look at that. Dr. Bennett, I see you were going to make a comment, and then I'm going to turn to Dr. Richmond. And in the meantime, medical doctors, I need you all to take a look at the comment that was just posed on the chat.
Hey, we only have one minority physician here, female physician. We should let Dr. Farhan chime in. She's young. She's working in the trenches with the COVID patients, and let her speak real quick.
Yeah,
Okay,
I was going to mention her comment, but I think it's probably more important if it comes directly from her. I was just going to
Okay. Doc, are you there? You want to raise that question? Okay.
She may be muted.
Is she muted? Okay.
She's having microphone issues. She's
Okay,
having
well,
microphone
it's
issues.
okay. Dr. Bennett, can you raise her question for us?
Yeah. Right. So I was talking about the phase 3 and phase 4 process, and Dr. Farhan mentioned that it's important to also add that, again, in the phase 3 process, you're also examining what we call effectiveness. You're trying to make sure that therapy is effective. That's a part of the phase 4, whether or not the intervention is a pharma drug, or if it's a medical device, or if it's a vaccine. So that's an important point, and it has to be done in a controlled environment. And so that's why in those particular processes, you want to make sure that anything that would essentially confound your variables in a phase 3, you have to make sure that all of those things are addressed up front in your statistical consideration of that particular trial. Also, Dr. Farhan mentioned that when you are moving to applying that intervention broadly to, say, for example, a phase 4, you're also looking at effectiveness and the performance of the intervention that you're trying to get approved, and how that happens sort of in the real world, process. So I just wanted to, that's Dr. Farhan's comment, and I just want to add some clarity to that since she's having some microphone issues.
Thank you very much. I appreciate it. And I'm being mindful of the time, but I am not going to allow this great conversation to get lost. Dr. Richmond, you're up. Dr. Bennett just did an outstanding job of breaking down those different phases, as well as the added dialogue that was provided by Dr. Farhan, who's on here. So now I'm going to get to you. And I want you to address this concept of detection versus inclination. You're providing us with some information about these two different forms and how incorporate what is happening when a person gets tested.
Thank you, Dr. White. So I'm going to share my screen with you guys to help you with the visual of what I'm trying to say as there are diverse populations in the audience. Okay, can you guys see my screen pretty clearly?
Yes, sir.
Okay, so to begin, what we're really talking about a lot of here is predicated upon our immune system. And our immune system is an around the clock or a 24/7, 365 surveillance system. It is constantly on guard, constantly keeping watch, and constantly scanning our internal environment of our body, as well as the external for things that may go wrong. It's looking for trouble, looking for signs of damage or threat. And so we have two overarching types of immune systems. We have the innate that we get from birth. We have this naturally. Everyone has this first line of defense. And so in this cartoon depiction here, you see a fence, and right here in red in the top left, we have our guys standing here with their hands up. Well, these are representatives or cells of our immune system. And what their job is to help reinforce or protect this fence. So some of the physical barriers that we may have are like our skin, tears, earwax, mucus, urine, and stomach acid. Those solutions, those barriers exist to help keep out bacteria, fungi, and viruses that may try to come in and attack our health and disrupt our well-being. Every once in a while, shown here in the second box, some bacteria, virus, or fungi do get through. So in this case, we're talking about the COVID-19. It gains access to our body, to the host, and it presents a threat to us. This is despite our immune cells shown right here in orange going around trying to take out the trash or get rid of the waste, get rid of these bad things that are in our body to help keep us healthy, just like we go to the restroom. When things become overwhelming, we have some cells, some specialized cells here that will call for help. Help is, and so when they ring the bell for cavalry to come, help comes in the form of over here, our adaptive immune system. And so when the adaptive immune system is kind of called or receives the call for reporting to work, they send mostly B cells and T cells. And so these specialized cells here are going to use chemical messages to recruit each other and tell more of their friends to come, hey, there's a fight, let's go. And once they get there, the B cells are going to mark anything that's not supposed to be there. So it knows who each person is that's supposed to be part of your body and what their job is and things like that. Anybody that stands out of place or anybody that seems foreign or not domesticated is going to be tagged to be destroyed. Then finally, the killer cell comes in and is in charge of that destructional process. So all of this is important because this is how the immune system works night and day, when you're asleep, when you're awake, when you're active, and in between. And so with that, I wanted to lay the foundation of, well, how do we get to actually finding out if I have COVID-19 or not? And so that's going to be done through testing. And it's important to understand that there are general tests that go on for maintaining how the status of your body is how your body's being maintained. But there's also testing when something seems awry or something's not okay. And there's a fundamental difference between detection, or which is actual screening, when things may be abnormal or something's off, it doesn't look like things are supposed to look, versus when there's actual diagnosis of things going on. When, hey, this is actually cancer, or this type of cancer, or this is actually just a scratch or just a cut, or this is actually heart disease. So with that, testing for SARS CoV-2, which is actually COVID-19, that's the specific virus, we have two types of testing that goes on, the viral test and we have the antibody test. And so these microbiology tests, specific to the viral test will allow a medical team to determine if you currently actively have the coronavirus in your system. So it will detect the virus itself. And we currently have tests that are in operation that can give you responses as early as 45 minutes or as late as 2 days, depending on the type of test that you take in the center that you receive your test. The FDA has recently approved, or to date has approved, two types of tests for the upper and lower respiratory tracts. One is a nasal swab, which is PCR based, and the other is a throat swab. And so that's taking something that we know looks like a Q-tip and sticking it all the way back into the back of the nose or into the throat to catch some saliva. And then we're gonna send that to a lab to be analyzed.
Dr. Richmond, and they didn't tell us how much that thing was going to hurt.
So they didn't tell you how much it was gonna hurt, but they did try to make it more affordable and applicable to the common person. But yes,
But it hurt.
It hurt. Yeah, it can hurt now or it can hurt later. It can hurt now or it can hurt later. So, and I mean, maybe that's a good point to stress that why it's important to get these types of tests, should you feel that you have a number of the corresponding symptoms. Things like this cost time and money and may be kind of embarrassing or painful, but imagine how much time, money it would cost and how painful it would be should you not take the corrective action necessary at the best time that you can. So complementary to this is the antibody test. This test will only tell you if you've previously had coronavirus. And so we'll start seeing more antibody tests roll out as they're being developed and proved efficacious. This test only detects the immune's response to the virus itself. So when the virus comes in, our immune system produces a serological or antibody response to take care of that virus or identify it. And this mechanism of testing will detect that response to the virus itself, to the RNA. So your results for this test can come in as early as 1 week or as late as 3 weeks for the ones that have been approved by the FDA. And so right now, the serological testing being used is based on your blood. So you would have to donate a portion of your blood, or a medical practitioner would have to sample a piece of your blood in order to screen it for any antibodies that may be present. And again, the question being answered here with the viral test is, do I have the virus right now? Whereas the question being answered with the antibody test is, have I previously had the virus? We currently have no test that will tell us if you will get the virus or how likely you are to get the virus. That's really based on your practice of lifestyle and the exposures that you have in your day-to-day.
Brother White, I'd like to get two of either Dr. Burchard, Dr. Bennett, or Dr. Richmond. There is a term, and I think they can define it better than I can, as herd immunity. And that's something thrown around, and I think it's very important that people know what that is. And I'm gonna defer to these brothers who would do a better job than me.
Sure. In general, herd immunity refers to the idea or the concept that the general population or the majority of the population has immunity to a particular biological threat.
Yeah, but it requires about 70% of the population have it. We use it for measles, and we're nowhere near that. So the current prevalence of SARS-CoV-2 in the United States is about 1%, so we're not close. I do want to throw in one thing. A lot of the NCAA Division I football players recently tested positive for COVID, and they happen to be all African Americans. So it's a warning to everybody that lightening the restrictions is risky, and the virus is alive and well.
Thank you.
Exactly.
All right. Appreciate it, Brother Richmond.
So yes, moving along. Well, how does this work? How does, for example, the genetic testing for this virus work? And so the PCR application, which the FDA just now approved, reverse transcriptase PCR, which measures the enzymatic activity. So as you present to a testing facility or a medical facility, a sample will be collected in the most painful way here, as shown with the cotton swab. Then RNA will be extracted from that mucosal sample, or that sample of mucus or saliva that has been collected from you. From that RNA compilation, there's going to be DNA that is copied from that RNA. So in this process, we're going to use biochemistry techniques to make DNA from the RNA. And once we have a complete strand of the DNA of this RNA of this COVID-19 virus, then we're going to copy it or amplify it. But when we're amplifying it, we're using a dye here that will be copied. Yes.
No, continue.
Okay, that will be copied when the DNA is copied, the DNA of this virus. So the way it works is basically a counting game or numbers game, quantification. The more viral particles that are copied, the more fluorescence or color that we see, or that our machines will be able to see once this DNA is made from the RNA. And then the results can be interpreted as positive or negative. So that's using the RT-PCR process. Another process, which is specific to the antibody process, has, in this case, by this company, 3 different examples, but they all work very similarly. So from a serological test or the patient's blood sample, a plate or a dish that has the antigen anchored, or that has the protein moiety of the virus anchored there or presents it, will allow antibodies from that patient's blood to bind to this moiety. And once it binds, it produces an antibody response. So the antigen and the antibody bind together here, creating this complex. Once that complex is created, a secondary antibody comes in that has been tagged with another fluorescent marker, or a marker that will allow us to see color. And then from there, we can quantitate, or we can quantify, or get a numerical representation of of how much virus you have in your system. And so with this test, it's predicated on a threshold. So if you have more than a given amount of virus considered to be a threat, then that's when you will be flagged, or your case will be flagged, and it's considered that you have tested positive for the coronavirus. That your antibodies here show that you've had coronavirus in your system previously. And that was a rough explanation, but please let me know if you have questions. So there are limitations, though, to the process of the testing, including the fact that it's time consuming. So even though at the earliest right now, we have tests that show results within 45 minutes, that still limits how many tests can be conducted in a day or in a certain segment of time to screen the overall population. To go along with that, the availability of supplies or resources. So having enough machines, test kits, or the equipment, or even the proper protective equipment for the healthcare practitioners to wear to protect themselves and their loved ones is a bottleneck for this situation. And then lastly, we have a level of concern of accuracy. So specificity and sensitivity. There's a large concern with the number of false positives and false negatives that are being observed with the different levels of testing that we're doing. So last I checked, there was a federal test being used for COVID-19 that our president was using or relying on, but it had a 15% inaccuracy rate. And so that's very high. That's saying 1 in 15 people out of every 100 tested is likely to have been diagnosed with the wrong status. And so if those persons are allowed to go under the radar, then you can have a second wave or tertiary or third wave of reinfection within the population. And so those are things, yes.
Yeah. No, I'm just saying, I love this. And like I said before, you're providing so much. I want you to continue because you're providing so much information. Again, like I said to all of the doctors in here, remember to kind of double back and hit that summary for those who are following you and those who are trying to keep up with you simultaneously. But as Dr. Dr. Rod said, this is outstanding discussion, and we have to keep moving and pushing to provide more clarity. So Brother Richmond, please continue your piece. And then Dr. Graham, I got a question for you based off of something he raised. So I'm gonna be right to you when Dr. Richmond gets done.
Dr. Richmond, before you continue, can I just highlight and underscore one fundamental point about your presentation as well? And this is around sensitivity. I believe one of the physicians on the line earlier in the discussion mentioned the FDA's EAU, emergency authorized usage, when you're developing a process by collapsing the time frame. Now, this is where this, the challenges for drug developers, because when you start doing that, what happens is that you rush a particular drug to market, and when you rush that drug to market, that drug is now, or in this case, an antibody test that is not sensitive enough because you haven't done the proper controls to be able to ensure that the test is sensitive enough that it can actually detect whether or not your immune system have over time developed an antibody as a result of you being infected in the past. And this is the challenge that the concerns as providers, you have to be concerned with. So I just wanted to add that bit of commentary, Dr. Richmond.
Excellent point. Thank you. Thank you, brother, Dr. Bennett. So moving along, as I prepare to wrap up, I wanted to touch on, so exactly how does the vaccine work? and it's predicated upon a normal, healthy, functioning immune system. So if over here we have this book, which represents basically our catalog, our voluminous representation or inventory of everything that we've ever ingested, whether it be a liquid or a solid, and sometimes things that have come in contact with our skin, our body, our immune system, our memory cells keep track. They keep a log of those things. So they keep track of the color, the shape, the size, the surface. And so, for example, here in this example here, a child eats a strawberry, the immune system recognizes it as food and absorbs it. But not only did it absorb it, it recorded that information for later. Again, the shape, the size, and the surface, so that it could recognize it. The important thing here is that should this immune system or this person come under attack later, then the immune system can sort through its entire inventory to say, hey, I recognize this, or hey, I do not recognize this. And if it's something that we've come in contact with before with, for example, if we've been inoculated against something, or if we've received a lesser exposure to a potential threat and our body was able to defeat it, our body now is able to recognize it for what it is. It can also send out a very prompt signal to say, hey, that's a threat. It means us no good. Let's go ahead and get rid of it. And they kill it right there. And so the sum total is that vaccines prepare our bodies to fight against harmful diseases. And so while we've had the results of the Tuskegee syphilis study and some other maladies throughout medical history, and with respect to apartheid, it is important to know that there are vaccines that work in our favor, and it is efficacious to have vaccines around. And if there's potential for a vaccine for COVID-19, there is potential for the vaccine to help us as well. So to take this a little bit further, do we still really need vaccines then? So if we have a garden and we're cultivating this garden, and it's a community garden, meaning that whether it's a co-op or whatever, we all pitch in to grow and maintain this garden. It's not enough to plant the seeds and fertilize the ground and water the ground and make sure it has adequate sunlight. So we must also mulch the soil. And in the process of mulching the soil, maybe we want to take turns. So without having a dedicated schedule, so people just show up on Monday, then someone else shows up on Tuesday, someone shows up on Wednesday. Well, after so long, maybe that schedule doesn't work anymore. So maybe on Thursday, no one showed up. And then on Friday, everyone showed up again because they wanted to get something in before the weekend. And then again, before the weekend, no one showed up. Well, now the weeds are going to overgrow in that garden. And we'll have this rolling cycle, kind of like a roller coaster, where we have a lot of weeds and we have no weeds. So weeds will be those threats, those biological threats to our body, the fungi, the bacteria, and the viruses that are trying to get in and take over. Again, they want a healthy host. So it's important that we get the clinical trial process right, that we don't rush the clinical trial process, that we do figure out what are our limitations, what are our advantages, where are our weak points, so that we can hone in on those and tweeze those out. So respecting the scientific process and allowing science to dictate what we understand and what we know, and us only interpreting what we find and what we learn from this process is extremely important, so that we can provide the best overall product for the patient population, which is us, and to promote good health. And so as a small caveat, what I usually tell patients or non-science individuals when I'm talking about cancer is, so if I'm diagnosed with prostate cancer and I have stage 3, or let's say stage 4 terminal prostate cancer, and it's highly recommended or suggested that I enter into chemo and radiation. Speaking to the chemo side of this, I mean, I really could just drink bleach. But the problem is while bleach is highly effective, it's also very non-specific. And so we go back to the specificity and sensitivity. So my cells will all be sensitive to the bleach and they will die. The problem with the specificity is that the bleach is not going to distinguish my healthy cells from my bad cells. And it's going to kill them all just the same. And keep in mind, bleach was very effective at what it does. So that clinical trial process is very important, that we respect the process so that we can get things right. Other things that Dr. Bennett mentioned earlier was enrolling patients into the trial itself. So while we do need a lot of African Americans, male and females, to join the appropriate trials, we need other demographics as well. So the cures that go out there, or the drugs that are labeled as cures, only work most effectively as a cure by that definition when they work on all people that it was designed to work on. And so as we're looking for something for COVID-19, it must be kept in the minds of the scientists working on these drugs or on these processes, that this is a virus that attacks all people, all ages, all demographics, all walks of life. And so this is a global issue, which is where the emergency use authorization comes from the FDA, saying, hey, this is something that's wrong. We need help, and we need help now. It is not the normal process for the government of the United States to be solely responsible for the development of drugs. That's not the normal process at all. And it's usually the pharmaceutical companies or the biopharmaceutical companies that are in the trenches developing these drugs, and they have a lot to answer for if they get it wrong. And so that's why they want to respect, that's part of why they want to respect this timeline for, how things are done. However, during these unprecedented times, we do have a limited number of individuals that are putting undue pressure on the scientists from the side of politics in order to affect the overall process and how long this takes. So getting back to this presentation, I want to wrap up with these bullets. So vaccines help your body identify what infections look like so that the immune system can use its natural defenses to treat them. Again, natural defenses. Vaccines do not change how your body acts when it encounters something. They just help your body identify what it encountered. So your immune system is trained to do very specifically what it does. And if you allow it, that's what it will do. So vaccines teach our immune systems to fight diseases that otherwise would take too long to recognize as harmful. They help us identify what is a threat versus what is a non-threat. If you take a bad guy and put him into a police car and let him drive around the city, he can cause a lot of damage. No one's going to really recognize him until it's too late because he's driving a police car. Versus if you put a good guy or a good woman in that police car and drove them around the city, they can do a lot of good or a lot of help. Vaccines are the best preventable the best preventable care we have to protect ourselves from disease. So with the predication of an inoculation, it's really just giving to the immune system a representation of a foreign threat so that it can learn how to defeat it. And should it recognize it later or should it encounter it later, it has a mechanism for defense. And lastly, I just want to wrap up here. So this is a very lame cartoon depiction of the coronavirus that we're dealing with right now. If you wonder where the name coronavirus comes from, so this outer coating right here is what scientists viewed under their microscope when they first saw the virus. So that's the corona. It resembled the corona of the sun. That's the co of COVID. And then you have VI for virus and then V for disease. 19 is when it was first presented. So COVID-19 is where it's first recorded, I'm sorry. And so that's where we get COVID-19 or coronavirus from, which is just SARS-CoV-2. So what you see here is this very simple organization of the structure with the RNA or the genetic material right here, nucleic acids. And then we have member proteins here and then spike proteins here, which the majority of therapeutics being designed right now are against the spike protein. And then we have the envelope protein. What's not shown here kind of in this yellow is a lipid membrane or a fatty membrane. And so my last message is it's important to wash your hands. So whether you have alcohol or hand sanitize, that's at least 60% of alcohol, or you have hydrogen peroxide, that's at least 3%, or you just have antimicrobial hand soap. It's important that you wash your hands all the time and common touch surfaces, because once those suds form and you're slishing it up for 20 seconds or so, the micelles or the chemical properties that are formed around these fatty substances in this virus holding it together are going to be disrupted or destroyed. And as this virus is soaked up and sudded up, the fats start to fall apart. Once the fats fall apart, the entire structure falls apart. Once the virus falls apart, it can no longer infect you. And so that's why it's important to wash your hands. And with that, I'm just going to conclude and throw it back to you guys. This was just a slide I threw in here earlier while we were talking. So this projection here of a normal, unrushed phase clinical trial here shows you well over 3 months or 6 months or anything like that. This is just a traditional process. And with that, I'd just like to end and jump back into all of us sharing.
I'm Dr. Richmond. I'm absolutely laughing. Excellent job. I'm laughing because I was looking at a comment that Dr. Wright said. He said that Brother Richmond. He said Johns Hopkins University offers a class on everything that you discussed, and he said, and Dr. Richmond just broke it down in 10 minutes. So excellent job. I'm going to go to Dr. Blanchard, and then Dr. Graham. Here is the question that I'm going to raise based off of what was said. You know, vaccines are built on different genetic profiles, and so I gotta ask you a question a little bit more, Dr. Graham, on that context of being built on genetic profiles. Dr. Blanchard, Bashard, I'm sorry, Dr. Bashard.
Oh, that was Dr. Richmond. That was the most awesome presentation I've ever seen.
Thank you.
On basic biology in plain English. And I just emailed your boss, Gary Gibbons, and Griff Rogers, who's the head of NIDDK, and David Wilkes, who's the head of the Robert Wood Johnson Harold Amos program, and his assistant, Nina Artery. That was the most awesome presentation. I'm going to hit you up for your slides, and then maybe even have you speak at UC-South and get you a job promotion. Thank you very much. Thank you very much.
I'm in the process. I'm in the process right now of actually applying for medical school.
That brother, Dr. Richmond started talking about the garden, the weeds in your garden. So again, one of the things that I love is that when you have this discussion and dialogue, and then have the ability to transfer that information. One of the entertaining comments I made to the group, to everyone, I said, the mother of the church during service, when the pastor is preaching, in the call and response manner, will often scream, "Make it plain, preacher, make it plain." And so I appreciate you brothers with the knowledge that is common knowledge to you, but not common to the lay people. So I'm really appreciating what you brothers are doing in transferring that information in a way that is certainly plain. So Dr. Graham, I just heard what was shared, the breakdown from what the others have done an excellent job of articulating. But I heard you mention before that vaccines are built on different genetic profiles. So I want you to take what was been discussed up to this point and incorporate that concept into this discussion.
I think it's like a number of the presenters already said. It highlights the importance that we have to be part of that sample size so that we are reflected. Our genetic makeups are informative to that process so that we don't get vaccines that are based on data that is derived from people that don't look like anything like us. So that is, in my sense, that's an imperative for people to kind of lose the anxiety, or at least reduce the anxiety of being involved in these trials. As the doctors told you, there is an impetus now to make sure that these trials are diverse. So that was the point. But I'd like, if I may, I'd like to just bring up one other point that kind of puts not a cap on, but informs this whole discussion, which was fantastic that Black men came together with this much information. So here's the thing. As of 5 May, 52% of coronaviruses and 58% of coronavirus deaths occurred in counties with disproportionately high African-American populations. That was on 5 May. To make this even more worse, the CDC just revealed that before April 15th, 78% of counties were not reporting racial data. So that first thing I gave you is even worse than we thought. And then the last thing I'd like to say is I'm a trustee of the ATS Foundation. We've gotten a chunk of money to do education to the African-American community, okay, about COVID and about the disparities. They're out searching a lot of ad agencies. With the FRAC's permission and the people on here, I'd like to give some of the archive files to the foundation to see, because what they're trying to do is come out with information that can go directly to the Black community.
Absolutely.
Well, my only caution is that Dr. Richmond copyright those slides. Seriously, because they'll be co-opted real quick.
No, I agree.
Well, we'll delay that.
Just make sure.
For Dr. Richmond. Dr. Richmond, it sounds like you might get you might be in medical school on the West Coast if you keep it up.
Right.
Dr.
Yeah.
Brother,
Go ahead and expedite that, Dr. Richmond, if you can. Dr. Burchard is a good friend and he's right on. You don't want to have that stolen, but by the same token, I want to see that get out to people of color. And the people I'm working with, the foundation, I've wrestled them after 20 years of being in leadership with an all-white organization to finally do something.
Beautiful.
Well,
Brother
I
Ken,
just sent it to Gary Gibbons, so don't worry about it. I don't have your slide, but I sent Gary and Griff Rogers and David Wilkes, who's a dean of UVA, a note about you. And contact me, email me after this meeting, and we'll get in contact.
I got you.
Dr. Dumas, as the public health expert, what's interesting about this great discussion, and I've been doubling back, is that your field and your profession, and the same with you, Dr. Wright, in terms of our profession, the social work profession, we have the responsibility of taking information like this and getting it on the ground level to the people. So Dr. Dumas, you're the public health expert. You are really one of the things that we know, that language and communication is the tool of the movement. So Dr. Dumas, how would you go about transferring this type of information to the masses?
I think that's an excellent question. And so as I'm listening to everybody, the thing I keep on thinking about is making sure that that we break it down to the most simplistic part. So, one of the things we get taught in medical school, in public health professions and in PhD programs, is really have to understand that people's literacy is probably at a 6th grade level. So really listening to that. But I think the biggest thing that, is really important is, reaching the masses, always utilizing those gatekeepers. And so as I'm listening to this stuff, I'm thinking about how do I package this when I'm training my community health workers? How do I package this when I'm training my physicians and nurses and lawyers who are in my executive MPH program? How do I package this when I'm going into the community? So right now, I'm just, like I said, I'm taking everything in and just make sure that I'm listening. So I know that Brother White's going to ask you once again at the end, break this down in 30 seconds, 45 seconds of what does this mean? Because, we have these people that are coming to us as physicians, as public health practitioners, as PhD researchers, whatever, to say, okay, you're talking all this stuff, but I don't understand a word of what you just said. And so I'm going to push that back to you, Brother White, as we continue on with this conversation.
I have it because one of the things that's, give me one second, Dr. Richmond. One of the things that's been riding me, and I'm gonna run to Dr. Bennett on this one. One of the things that's been riding me, just really, and Dr. Richmond did it a little bit, but I want you to touch it a little bit more for me, Dr. Bennett, in terms of understanding antibodies. Even though Dr. Burchard talked about the percentage of this herd community concept, but if you don't mind, just give us a little bit more clarity in terms of understanding antibodies and this whole concept of the herd community.
Yeah. So, I think Dr. Richmond actually laid it out quite nicely, in the animation process, a part of his presentation around why it's important for us to be able to detect antibodies, right? Because we have them since birth. If there's an incident that happened with you as a baby, if you've had any type of allergic reaction to a bee sting or a wasp bite or food and those kinds of things, your body will maintain those antibodies for the rest of your life. That is how sophisticated the human immune system is. It is constantly churning. It is constantly working. Think about, many of you have seen, I forgot the movie where The Matrix. Think about The Matrix. Think about The Matrix, where you have those machines that are running through through this particular system, searching for things that are foreign, that should not be there. And once it finds it, it destroys it. That's what the immune system is designed to do: find foreign things that does not belong here, and it destroys it. And so, Dr. Burchard mentioned earlier that the whole concept of herd immunity, you need to have a very large subset of the population who have had exposure to the virus in order for you to have what is called herd immunity. The terminology herd is generally reserved for example, goats and cattle and those kinds of things. But in medicine and science, it's applicable because in a case like this, you really need to ensure that the masses have actually been not necessarily exposed to it, but you want to at least understand who have been exposed to it, which is the reason why you want to develop your vaccine to be able to test it. But again, those testing process or the mechanism by which you are utilizing the test needs to be sensitive. Sensitivity is the pivot. If it's not sensitive, you run into those false positives and false negatives, and that in itself is junk. All right, it is literally junk, because you don't want to go into your physician's office and the question that you may pose to your provider is that, what's my chances of essentially being inoculated against this virus? And when the doctor may look at you and say, well, you have 1 in 15. That is not a test that you want. That is not a vaccine that you want. That is a pretty slippery slope of practicing medicine. And so, you know, I'm not entirely sure if I can assess that any more simpler than how Dr. Richmond did, but that's sort of the concept around antibodies, because it's one of those things that must be detected, but the only way to do it is if you have a sensitive test, but that test needs to have gone through a proper evaluation, a proper trial process. It needs to include a diversity of patients. It needs to confound for all the different stratifications that are needed in order for you to be able to utilize that particular tests in the clinic.
Thank you. So now we are going to, as we move, we've gone over, but we expected this. I was literally, by the time we got to the point of the vaccine, the shot in the dark part 2, I was literally prepared for the added discussion. We're going to go into our myths, and we're going to go into our myths and our facts, but I want to hear from Dr. Richmond first, and then I'm going to ask this question that Brother Dumas posed in terms of, you know, what do we do right now with regard to helping patients understand the vaccinations, and if we are saying that vaccines take time to develop, how are we communicating that? So I'm going to, with the final discussions, I'm going to double back with all of you on that. Dr. Richmond, and then we're going to get to the open questions about the myths and facts.
Yes, 2 things. So first, I just want to address the question about what do we do right now, since we don't have a vaccine that's available right now. So physical distancing is very important. It's not a game. It's not there just for play. If you don't have to go somewhere, and particularly be in groups, then don't go. It's okay to stay home. Make sure you're protecting your mental health. That's important. You could also potentially save some money. The other thing is washing your hands and commented surfaces is extremely important. We cannot see this virus with the naked eye. You can't see the potential threats. You can't see transmission. So we aren't that far yet. So I just want to say what we can do right now is work on prevention. That's very important. That's pretty much the best thing, in my opinion, that we can do given our current circumstances and the fact that we were behind the ball in the first place. And my other statement, I just want to say, so Dr. Farhan says that she has in the field experience that she wanted to share real quick. So I was going to see if we could try her microphone again.
That is so funny, because she and I have been texting. So I appreciate, you know, you medical, you doctors, y'all like a fraternity within a fraternity. I'm smiling the way you all are giving each other alley-oops and stuff. So, you know, that's, but it's great. And this is truly what iron sharpening iron looks like, even in this discussion and dialogue. Dr. Farhan?
I'm here. Can you hear me?
Yes, go ahead.
Okay. I'd like to thank you all for letting me join, and just say that I've learned so much listening, and that these, you know, I think the value and the real power in these groups is exchanging information and then disseminating that information, right? That we all have communities and networks with which we can reach out to. And so I just wanted to speak to that for a minute. I feel like the point has been very eloquently made that we are a long ways from a vaccine, and that for that vaccine to be effective, that we need representation by clinicians, by scientists, by trialists, and by patients. And that requires really getting that message out. But in the meantime, our communities are dying at alarming rates, right? And so you know, planning for a vaccine and making sure we do the work is important. But in the meantime, there is a ton of work to be done on the journey till we get to that vaccine. So I just wanted to speak to that for a moment, because I had the opportunity to volunteer yesterday in District 10, which is the historically Black community in San Francisco. And the event was hosted with a number of Black community organizations, and there was music, and there was free food, and it was free testing. It was, you know, it was the antibody testing, and it was the RNA testing. So, and it was patterned after events that had been done elsewhere in the city to really reach more marginalized communities. And it was unbelievably disappointing at the turnout. You know, they were expecting much, much higher levels. A lot of effort had been made, and people did not come. And it got to the point where they actually asked for physicians to volunteer to go out into the community. And so I did that. I went out with a couple of community outreach workers, and we went to the most marginalized parts of the city. We were, you know, approaching tents for testing homeless patients. We were knocking on doors and ultimately reaching people that way. But I think it is so reflective of the impact that deep distrust of medicine and hospitals can have on being able to get testing and interventions to the communities that need it most. And so that is where the effort needs to be made, because if we can't figure out how deeply affected someone, if we can't count the people that are infected, if we can't count the number of people that have been exposed, then they aren't counted when we're trying to figure out where to disperse dollars and where to focus attention to fight the virus. And so that is the message that I think we came away from at the end of the event, there needs to be more work put into explaining why this is important. That being counted in this sense is as important as being counted in every other you know, aspect that we're talking about right now.
Beautiful. Thank you very much. I appreciate you, Dr. Brand. I'm laughing because I got like 2-3 different private texts to make sure that I had you speak. So that's why I was laughing about the beauty of you all definitely kind of chiming in. So as we move towards the end of this dynamic historical discussion, I want to pose some questions about some myths and making some distinctions with regard to some myths and facts. So I'm going to ask some basic questions and certainly invite any of you to kind of chime in. So the first question is very simple. Are the vaccines safe? Once they come out, can we be comfortable, given what you all have just articulated in an outstanding manner, that we are in unknown territory? So then back to the original question that Dr. Bass had to address around anxiety and fear last week. So are the vaccines safe when they do come out?
So I think, Dr. Bouchard, you're probably not going to disagree with me on this one. Safe is a relative term in medicine, right? Because I have yet to review a safety profile of a particular marketed drug, and there isn't some form of what you would call it, some side effect profile that essentially that a particular product may essentially exacerbate in a patient or whoever takes the drug. So it also, it's sort of the real question that you have to ask is, does the risk outweigh the benefit? And so if the risk outweighs the benefit when you're taking a particular therapy, oftentimes you're probably not going to get an approval from the FDA to be able to put that drug to market so you can actually have patients on it. You do have some therapy out there, some maybe in the form of vaccines, that do have some pretty powerful side effects. But again, the benefit of taking the therapy far exceed the risk of you being able to essentially take that therapy. And so essentially the FDA will, give you that thumbs up. So I think, safe is a relative term, just sort of depending on the side effect profile, the profile, the toxicity profile of that particular therapy. In vaccines, I have yet to review a safety profile of any vaccine that does not demonstrate some components of it that, so for example, for the non-scientific and the non-medical person, when they read that, they look at it and say, oh no, I'm not taking that. You're gonna have diarrhea and all this other, no. But in medicine, we recognize that these are side effect profiles that were demonstrated as part of clinical trials, but we have to make sure the people are aware of them. That doesn't mean that you may have those same types of things, but we want to make sure that you address them. So
Powerful.
This is Dr. Grant. I'd just like to add a comment. It's personal. Not supposed to give personal things, but I'll give you an example. 30 years ago, I received an artificial aortic valve. Okay. I take Coumadin. Coumadin is warfarin. It's rat poison. Okay. I take that, and clearly the benefits of me not clotting off that valve over the last 30 years outweigh the risk. Now, I do the right things in terms of monitoring myself. Now, we're not supposed to give personal examples, but I think that's the way we have to look. We have to mitigate risk on one hand, but we also have to protect ourselves on the other. And what the previous speaker said, I think the challenge for us as not just providers, but people who speak in the public domain is to make those points intelligible to people, okay? Because they're always gonna hear the bad, but we don't balance the good, and we don't have that discussion of a risk-benefit analysis. So sorry to give a personal example, but you know, I've been taking it for 30 years. I rode 15 miles on my bike yesterday, so all good.
I appreciate you. I'm actually looking through my notes trying to figure out when in these 11 weeks that I say don't give a personal example. Dr. Wright and I, we're social workers, so we understand the benefit of this thing called use of self to connect with the client. So definitely, I'm wide open with that. Question number 2. Very quickly, there was a great point with regard to the Ebola. Doc, you want to raise that that question that you presented on the text?
Sure. Hi, it's Dr. Esteban Burchard. You know, again, I want to reiterate that we've had HIV since the 1960s, and we don't have a vaccine. And then we've made tremendous strides with polio vaccine. Those are some wins, but with the Ebola vaccine, it actually made the disease worse through this thing called antibody-dependent enhancement. So, you know, the president, who's a member, a game show host, has no idea of science promising a vaccine in 6 months because he's up for re-election in November is false. It's false. It is totally false. You know, we have to be slow and cautious. And again, I want to thank Dr. Richmond and Dr. Bennett for explaining the timeline, because we may have a vaccine, we may not, but it's not going to be in 6 months.
Thank you very much. Doctoral candidate, Brother Kyle Bryant, you just raised, you made an excellent point that I want you to kind of restate to the group. That you put it on the chat, but restate it to the group, and then I'll double back on that. Go ahead, Brother Bryant.
Yes, I just wanted to make the point that when engaging communities, particularly our communities, concerning talking about risk, usually I look at it from the standpoint, not from the medical field, but from environmental hazards. But it's the same level of discussion when you're talking about a hazard of any type. And our people tend to weigh factors such as the cost of treatment, or if it's going to be any cost, like I would imagine in the medical field, it would be whether the prescription that for this new drug you're offering costs more than what they're paying to treat what they already have. And often they will weigh the value of whether the new drug gives them more side effects that they would be uncomfortable with, and say, well, like say drugs for smoking cessation, they might just be fine with the side effects from just smoking, as opposed to taking a Nicoderm patch, which comes with 3 or 4 more side effects that they may be uncomfortable with. So that's how our communities usually look at those things. And so we have to find a better way to communicate value for the new drug, if it is indeed something that is going to be beneficial to them ultimately.
Thank you very much.
That's an important point, because again, those are sort of those false equivalents that you have to make sure that you stem them from the very beginning, simply because when you, as a clinician, you really need to dive into the, you really need to get into sort of the long-term medical impact, all right, to your system. Say, for example, an example that you just gave, versus you taking a Nicoderm patch, if you will, because it has those side effect profiles. But the thing is, you're at such a higher risk of lung cancer and everything else when you're doing smoking. So you have to balance sort of the long-term risk, the short-term, what I would call discomfort of taking, say, for example, something that can take you off of that ledge, so to speak. And I think once you start explaining ways to patients that they can truly understand and appreciate, this is a long-term thing. It's not something that is going to fix itself overnight. So if we can explain it that way and grasp their attention in those moments, so they can understand, do I want to risk it all by going full force with continuing to smoke, or am I willing to bear the, in some cases, short-term consequences of taking a therapy that can extend and prolong my life? Now, that is a thing that needs to be addressed there.
Powerful. So one more question, and then I'm going to do my round robin. You're going to have 10 to, you're going to have about 20 seconds to respond to my round robin questions. But I see a question that I'm going to present to you, Dr. Wright. The role of the social workers. Dr. Brand did made an excellent point about their experience in San Francisco, and I get to say in my sister-in-law's community, and the turnout. So certainly we now see that there is, that turnout can be reflective of a lot of things. But one thing we do know, which is in regard to what Dr. Bass said at the beginning of this discussion, it has a lot to do with the comfort. Who's there? So social workers, what is our role in aiding and engaging the community to participate in the clinical trials, participate in the vaccinations? Because while we have the Tuskegee experiment, the other part that we missed out is there was that nurse named Miss Evers, and that's who those men trusted. And so it's not always the doctors that they're going to have the trust, but it's going to be that person they see on the daily basis who walk with kings and keep the common touch. So Dr. Wright, what's the role of social workers in aiding in that process?
Well, Dr. White, I think one of the roles obviously is understanding who the credible community messengers are. And I say that because sometimes as professionals, this is not built from the outside in, but the inside out. And part of that community assessment is really identifying those individuals who are seen as credible, and then partnering or allying with them to then convey the message that was mentioned by Dr. Richmond and Dr. Bennett and others. Because without that, you still have to address the issues of people in terms of people's health belief practices and how they perceive or interpret what is being offered to them. Because if you don't meet people where they are, then you're going to have low turnouts. You're not going to have full engagement or investment in what you're trying to do. So that's a key part, I think, in identifying who the credible messengers are, and then helping to train them to bring the message to the level where people get it, as we said it before. Keeping it simple, not overusing terms that people do not get, because therefore individuals then are turned off from that.
Powerful. So I say to all of you all, there is a gatekeeper in this process, and certainly as social workers, I often make the case that we're the Green Beret of social change. We're the foot to the ground professionals, and so certainly to bridge that and facilitate that level of comfort. We are always going to make a case of understanding the role of social workers in that process.
Dr. White, can I just, I'm sorry, I don't mean to cut you off there, but I think there's one pretty popular myth, if I may, hopefully I'm not upstaging you here. So there's one prevalent myth that I think we just probably need to just give father to for maybe at least 1 minute. And is that in our community, there is this notion that if you're being inoculated against something, we, the medical professionals, are giving you a virus, right? So you have a lot of people who say, oh, I'm not taking a flu shot because you're giving me a virus and those kinds of things. So, you know, one of the things that we need to make fundamentally clear is that, you know, again, vaccines are immunizations. They are designed to stimulate our system to produce antibodies. So when someone develop a vaccine, they're developing something that mimics the virus. It is not an active agent. It is not an active agent, and it's not introducing a virus to your system, an active virus to your system. So it's an element that essentially helps to mimic what the actual virus would do, and then it stimulate your immune system to produce an antibody. And that's what happens in a scenario like that. So this whole myth around we're giving you a virus, there's no truth to that whatsoever.
I love that. Thank you very much, Dr. Bennett. Again, this is why we celebrate what you all are doing, because everyone who's taken the time to watch this have benefited greatly. And hopefully, Dr. Bass, those anxieties and those fears, we've managed to kind of mitigate those a little bit. So as we go towards the end, here's my round robin question to each of you. First, I'm going to ask you, you know, if there's 3 things that you need the people to know about what you shared, what are those 3 things? And then number 2, what do you say to a patient who says, should I participate? The 3 things you want people to know, and what should you say to a patient who's asking you, should they participate in the vaccinations and the treatments and everything else? Dr. Dickerson, I'll start with you.
Okay, what I would basically like to say to the patients and everyone is just to do your research. And as we discussed last week with the Hippocratic Oath, part of the Hippocratic Oath was to do no harm. And so thus, when I'm talking to my patients, I want them to do their own independent research about getting this vaccine due to the circumstances of the sensitivity and the testing that's out there, do their own independent research. And I don't want it just to be in my hands. However, if it's something that, whether they work on a job or a hospital, in the hospital makes them get it, that'll be a different situation. But I want to be their own independent decision. I don't want to do no harm. I want to uphold my Hippocratic Oath. That's
Beautiful. Thank you very much. Dr. Graham, those 3 pieces that you want people to walk away with, and what do you tell your patients about participating in the trials and the vaccinations?
First one is yes, they should appear, they should participate, they need to be representative in those. The 3 things I would like was what came up before is that this COVID-19 is disproportionately impacting people of color. We cannot afford to be the canaries in the coal mine. So we are the ones, when we see majority people disregarding social distancing, disregarding masks, that is not for us. We should stay prepared, as a number of speakers does. So when you're out 6 to 10 feet, when you're going into crowded things where you can't avoid closer contact, and I would generally say any traveling away from your home, mask up.
Thank you very much. Dr. Bennett, those 3 things you want people to walk away with from what you shared, go have at it.
Sure. So the 3 things that I would underscore from my discussion today, talking about the clinical trials development process, is that patients need to understand that it's a process. Not only it's a process, it's a long process. Not only is this a long process, but it's a judicious process. It needs to be a decision between them and their provider as to whether or not that trial would be appropriate for them to be enrolled in. And oftentimes it's incumbent on the clinicians to tackle myths up front with patients. You know, let patients know you are not a guinea pig. No, we're not going to inject you with some live agent that's going to cause harm to you. The idea behind a trial is there's not only the benefit to you as a person that is participating in the trial, but there's also the altruistic benefit that you may be helping to create a therapy that can save someone else's life down the line. And so if you can essentially point out those kinds of things to the patients, I think most of the time patients are very receptive. In addition to that, you also want to ensure that when you're talking to patients about a trial, you speak in terms that patients can understand. We always say if you're doing an informed consent, make sure it's at a 6th grade level. That's why it's written at a 6th grade level. And oftentimes we use technical jargons that sometimes it can throw a patient off, but you also want to make sure that you're not coercing patients in joining a particular trial. But you also want to balance those risks and the benefit and what those altruistic benefits are for that particular patient.
Thank you very much. Powerful. Dr. Richmond, 3 points you want to highlight from what you shared, and what kind of conversation would you have with the client? 30 seconds, go.
Okay, so first, my conversation with the client. All of these things that we're promoting to practice are great and necessary, but keep in mind they are half measures. So you can't just pick one and follow that. You need to practice all of them all of the time. So for example, if I live in Atlanta, Georgia, and I'm going to Piedmont Park or the public park to hang out with my friends because I've been inside for almost the last 3 months, that cloth mask minimally is going to mitigate or protect that person I'm talking to. It's going to kind of limit the chances of, if I have the virus and I don't know it yet, it's going to kind of limit the chances of me giving them that virus, for me transmitting that virus to them. It does not protect me at all, and it does not fully protect them. The best chance, if I'm going to be in close quarters, is again the N95 that is worn properly. So you guys notice I used to have a big beard. I've shaved my beard down just so I could actually fully wear my N95. I know doctors right now that are on the front lines that are wearing their N95 and then they're putting a cloth mask on top of it in order to prolong the use of that N95 because we have such a shortage of availability. So I'm not advocating that you go out and take out all the supplies that our first responders need, but I am saying that you need to practice physical distancing, washing your hands, and all the other prescribed processes. I'm going to stick to alliteration for my 3 tenets to hit. I'm going to say vote, volunteer, and verify. So you need to judiciously be involved in the election process, not just the national scale, but also your local scale. If you want change, if you want your voice to be heard, what you do locally counts and matters, and who you put into those elected offices actually matters. And I'm not going to argue if you believe it or not. Next, I need you to volunteer, not just for the clinical trials, but volunteer in your communities. If you have people that are at risk, you can volunteer to make a consolidated trip on a day you go to get your groceries. There are fewer people in the stores, and you can drop things at their door so that they can come out and get them at a more appropriate time or whatever. Remember, you are stewards of your community. Be the leader that your community needs. And last, I'm going to say verify your data. Check the sources of where information is coming from. Does this information hold up in other sources as well? All too often right now, our news sources like to be first, and being corrected is not the main goal anymore. So just because you hear something, don't just run with it. Be careful about spreading propaganda that may push this message or push some type of fear that's not necessarily true or doesn't stand up. We have a lot to combat, and we don't want to add ourselves to each other to that list.
Thank you very much. Appreciate you. We're making ground. We're moving, Dr. Bouchard. Your 3 points you want to share, and what would you tell your patients? 30 seconds.
Well, I just checked my 23andMe. I'm 5% African, so I hope I qualify as a brother. But I spent my time wrestling in college, and I was an NCAA athlete, and I want to tell you that many of the football players just tested positive. For Division I athletes tested positive this year, just this week actually. So the virus is alive and well. And African Americans are the most highly affected population. It's like 10-1, and be on guard. So that's what I would have to say. There's a lot of politicization of this. Don't believe the media. And I think that Chat with the Frat is important and should get more widely disseminated.
Beautiful, beautiful. And certainly Dr. Fran made her points where she kind of similar what others said. Number 1, socially distance. 2, mask up. 3, wash your hands. 4, get tested. And if you have symptoms, do not wait to seek care and actively engage in medical care. I'm going to finish this last question here because there is yet an elephant in the living room. So this last question I'm going to send to our public health expert, Brother Dumas. Here we are in the midst of protesting all over the country. We were already, before the protests erupted, concerned because states started opening up. And there were those who were even protesting in the medical field and abroad about states opening up. But then Floyd hit, Aubrey hit, Taylor hit, and it got people out in the community. So what concerns may we have and seek to anticipate by, number one, these states that are opening up, and number two, protest participation as it relates to new cases?
I mean, that's a very good question, brother. And so one of the things that, we're hearing in the public health community, just was on a call last week with the Department of Public Health for the state of Georgia. And so the numbers are going to go up. The spike is going to happen. There's been this increase in getting more funds to actually hire more contact tracers and case managers to really help with these numbers that are about to spike. And so is these people are calling out to the individuals that either testing positive or that have been around people that have been testing positive. It's important to make sure we answer those phone calls and actually do what they say. And so I was having a conversation with Brother Dr. Wright about, the impact of the social work community, and that, a lot of these individuals don't want to do certain things as far as isolate or quarantine because they have lives to live or whatever. So there's this conversation between us as public health practitioners and social workers to help them to finalize, get those sources and those resources that are really going to be needed. So once again, I think, Dr. Farad kind of talked about, the social distancing, the masking up. So if you are going to these protests, you have to mask up, making sure that we're washing our hands, making sure that all the people that have been in these protests, make sure you get tested. I know we talked about that, this thing hurts, but you have to get tested because you are putting other people at risk. And, we want to make sure that we're protecting the health and well-being of everybody around us, not just ourselves. So I'll end it at that.
Thank you very much. Gentlemen, brothers, I cannot tell you.
Before you leave, can I just say one thing real quick?
Yes, sir.
I wrestled in college, about 10 years, NCAA wrestler, and I've been choked out. And I understand the heat of the moment and testosterone that goes with George Floyd. But, I pray at night and I feel so sorry for Ahmaud Arbery to be hunted down like an animal. And I think that the whole story was missed by the COVID and the scandal. And I want you to pray especially hard for him for being just hunted down like an animal. It's ridiculous. I'm sorry to hijack that point, but I've thought a lot about it. I've been choked out. So like George Floyd, I'm sorry, but Ahmaud, I feel terrible about.
I appreciate you very much.
I've choked a lot of people out myself.
Well, I appreciate you a great deal. And I wrestled in high school in my first year of college, so I understand. But gentlemen, I'm going to tell you all, I can't thank you enough for the information and dialogue and discourse that we engaged in on today. I need you all to know that we made history. We made absolutely history because hopefully, Dr. Bass, somewhere out there, people are more comfortable. Hopefully, as we continue to repost this dialogue, that people are able to tune in and study and get their questions answered. For all of you, as we move through this, we see an outstanding process that has taken place on today. We had Dr. Bass open up by recapping around issues of anxiety and concern. Then we had Dr. Graham break down the difference between vaccinations and treatment, but that was not done by the work that Dr. Bennett did in breaking down that 3 to now we know 4-step process in the clinical trial, making it realistic that the promises that are being made around vaccinations is not realistic. Then we love the outstanding slides that Dr. Richmond provided us, and as a matter of fact, that might have even got him in medical school. You let anybody else tell it, but he broke down that process of detections and inclinations and made a significant difference. I'm still running those weeds in those diagrams through our mind because, again, like the elders in the church say, you did an excellent job of making it plain. Dr. Graham, double back again. and followed up with the discussion that everybody shared in terms of understanding the significance of the genetic profiles and what that really means, and why he has from day one made a case for why we must be involved. Brother Bennett doubled back and feel more of that discussion and dialogue around this concept of herd immunity, and helped us put that into perspective. And we made it all the way around to addressing some of the myths that were out there, and what we can even potentially anticipate, as Brother Dumas has shared, in terms of the role of protesting, to the outstanding points that Dr. Wright made with regard to the role of social workers and the formal and informal communicators. And so we've made it to an end. 2 hours, the longest one yet. 2 hours, but the information, I'm going to argue, is absolutely invaluable. So on behalf of the interdisciplinary team, Dr. Dickerson, Dr. Dumas, Dr. Graham, Dr. Bryant, Dr. Wright, Dr. Bass, and myself, we can't thank you all enough. On behalf of the Brothers of Alpha Phi Alpha Fraternity Incorporated, Omicron Phi Lambda chapter, it is indeed an honor and indeed a pleasure that we were able to bring this forward to you. Now, so I'm going to encourage everyone to go to South Fulton Alphas, our Facebook page, and look for the link to this, share it, like it, so that we may keep this discussion and dialogue moving. Once again, my name is Dr. Gary O. White, a licensed social worker, scholar, practitioner, and activist. And on behalf of the Brothers of Alpha Phi Alpha Fraternity Incorporated, Omicron Phi Lambda chapter, the South Fulton Alphas, also known as the Renegade chapters, we thank each and every one of you for joining in and engaging in this outstanding dialogue. I say to all of you, we are in history, we're making history so that we won't be history. So thank you all, take care, God bless, and until next week, we'll see you. Brothers, throw them up. You all take care, God bless, and I'll see you soon.